Very warmly welcome, everyone. My name is Anna Ridderstad Wahlberg, and I will try to moderate this. So today we have two presentations. One is from Per Svensson, who will talk about computational pharmaceutics and robotized screening. And the second one is from Swapnil Chavan, who will talk about automation of predictive toxicology by AI.
Yes, so my name is Per Svensson. I have about 20 years of experience in pharmaceutical industry, having worked at companies like Sanofi Aventis, AstraZeneca, and now RISE. I have also long-term collaboration with KTH in Stockholm, and my expertise lies in computational chemistry, robotized screening, material science, structural informatics, and the utilization of synchrotron techniques. So my talk today, I'm intending to provide you a condensed overview of our efforts within computational pharmaceutics and robotized screening in pharmaceutical development. Short outline. First, I will provide a concise overview of the computational pharmaceutics landscape. Following that, I will highlight practical examples in some key areas that showcase the potential of computational pharmaceutics. And next, I will present some of our efforts in robotized screening aimed at speeding up the discovery of novel materials. And finally, if time allows, I will share some personal insights in the area. I will focus on the product development phase and traditional small molecule APIs. I will not cover the drug discovery phase, and ADME predictions are not covered in this presentation as well. Here we have a basic overview of the computational pharmaceutics operational landscape. It covers everything from liquid crystal to bulk systems, where the solid state system dominates. If we look into the crystalline system, there is a vast amount of computational supports with many areas, including a range of in silico screening methods. For amorphous system, there also exist numerous amount of computational support, and here also you can get insight to kinetic properties. For both particle and bulk system, there are also numerous areas where in silico support is available, however, maybe a somewhat lesser extent. But overall, there is robust computational support accessible. And I have selected some examples from these areas to illustrate the computational pharmaceutics in practice. But before going into practical examples, I would like just to have a brief overview of the computational support available, which utilizes various levels of theory, ranging from quantum mechanics, molecular mechanics, as well as coarse-grained models, along with some statistical and mathematical models. And these methods covers a very wide range, both in time and length scale, and as a result, also a wide range of properties, as can be seen on the right-hand side of the slide. And instead of categorizing computational pharmaceutics by system investigated, as in the previous slide, we can also divide it into a more general way into three application areas. And this covers property, formulation and process optimization, analysis and interpretation of experimental results, supporting, for example, spectroscopy, powder diffraction, or NMR, and in general, of course, problem solving. Let's shift our focus to practical examples where computational pharmaceutics has been applied. This first example deals with crystal growth in connection with absolute configuration determination of an oncology substance, Vakinol, which is active against a very aggressive type of brain tumor, glioblastoma. In this collaboration project with Karolinska Institute, we were targeted with the determination of the absolute configuration of the active substance Vakinol, since it was unknown. There was, however, a small amount of diastereomere substance available, making the crystal necessary for the X-ray determination of the absolute configuration. Subsequently, a critical question emerged. Which solvent should we be using for the crystal growth experiments? And of course, with 50 milligrams of substance, you can't waste, and you have to be trying to target the right solvent at an early stage. And here you can see how the crystal looks initially, and these are very small and clustered and not appropriate for absolute configuration determination. In order to facilitate and support the experimentation, molecular mechanics-based solvent screen of the API was conducted. And here we can see the results for such a prediction. On the x-axis, we have the interaction energy, and on the y-axis, we have the so-called packing index. Each point corresponds to a solvent examined. The highest ranked solvent for the API was used in the initial crystallization experiments. And here we can see the results, and on the right-hand side, we see the result of the crystal growth, and this single crystal is mounted on nylon loop. And in principle, at least 10 times large crystals were obtained. Furthermore, we determined the crystal structure, and here you can also see that actually the solvent was included into the lattice, meaning that we got a solvate of the API. And of course, we were able also to determine the absolute configuration of the substance. Next example is connected to form selection and co-crystals, which is an alternative to salt-based APIs, and where a neutral co-former is added to the API to form the co-crystal. The question is, how can we find appropriate co-formers? Since this process can be very time-consuming, computational support would be extremely helpful to guide the discovery of interesting co-formers. By utilization of a molecular-based method, we could screen a library containing about 1,200 co-formers against the API, and in this case, it's theophylline. Here we see the outcome from the screening, and the highest ranked candidates marked with a circle in the upper left corner were investigated, and here each point corresponds to a predictive co-crystal system. And here, in the same way as before, the x-axis is the interaction energy and the y-axis is the packing index. And the 4 highest ranked candidates was experimentally evaluated, and 3 resulted in co-crystal formation. An example of a co-crystal formed with salicylamide can be seen on the right-hand side. It is noteworthy that the library also contains co-formers that is not suitable for pharmaceutical development. However, the inclusion allows for comprehensive understanding of what can trigger co-crystal formation in different systems. And an example of such a library candidate in this case is cyanophenol. And in this case, we were not actually able to get a co-crystal with this co-former. Next practical example deals with intrinsic solubility and solubility improvements. And if we look at the solubility from a solid state point of view, there are 2 factors affecting the solubility. The entropy that have a positive effect of the solvatization, while the interaction energy has a negative effect. So what can we do to reduce the interaction energy to increase the solubility? The solution that we introduce is a methodology where we perturb the packing or key interaction in the crystalline state to reduce the interaction energy. And here is an example of a perturbation prediction where different perturbation have been applied. And here the overall aim is actually to reduce the difference between the amorphous and crystalline solubility. And in the box on the right-hand side, we can see different perturbations. And the result, as I said before, is shown in the plot. One example of a perturbation could be, for example, exchange of hydrogen to fluorine. With this approach, it's really aimed for so-called brick dust compounds, which has a high melting point. And in principle, you can achieve up to 20 and even more times improvement of the solubility with this method. I will just show you a very basic example here of the perturbation approach on the benzodiazepine series, where we could show that it's possible to predict the effect of different perturbations of a larger series. The perturbations are marked with circles, and going from compound 1 to 4, there is a solubility improvement of about a factor of 10. This has been published some time ago, and if somebody's interested in the references, please don't hesitate to contact me. The perturbation methodology can be used also in the reverse to promote crystallinity, and in this case, we want to increase the interaction energy. Well, I don't have any examples of this, but it's the same principle, but we really work to find more crystalline material instead. Let's go to another example, which is dealing with surface inhibition by impurities. And in this example, there was seen a deviation in dissolution rate between 3 different batches. But we couldn't understand why. The batches was of high and similar purity, but in one of the batches, there was actually a very small amount of impurity that had a very similar molecular structure compared to the API. With MD calculation, it was actually possible to discover that this extremely low concentration impurity have the perfect fit in the surface of the API. And this would, of course, have an impact of the dissolution rate. And here in the figure, you can see the impurity fits very well into the surface of the API and also interacts very well. And we also made further studies of this case, and we could actually see that the impurity formed a solid solution, meaning that the impurity actually incorporates into the crystal lattice. And this is, of course, a very extreme situation and can create a very complicated situation. Finally, I will show 3 more general shorter examples of utilization of computational pharmaceutics. The first one is connected to the morphology and size of the API crystals that have an impact on many critical parameters, especially particle and bulk properties. Therefore, insight in how to control and manipulate the morphology can be very useful. And here I just show you a very basic prediction of the morphology, and on the right-hand side, you can see that it's at least a reasonable coherence with the actual crystal formed. The second is an example related to mechanical properties and how it is possible to track when and in what direction the mechanical properties can have issues, and where the API structure can collapse under application of external pressure. This capability can be very helpful in risk assessment and process development. And here is mentioned the Young modulus, often denoted as E. It is a measure of the stiffness and elasticity of the API and material investigated. And finally, a brief example of how varying mixtures of API and excipient can influence stability and solubility. And here we see the MD calculation of the API and excipient in liquid state and without the solvent included on the right-hand side. And in this case, by doing analysis of the hydrogen bonds and the conformation and a conformation analysis, it was seen very useful in understanding the system's stability and also the effect it had on the solubility. Having discussed computational pharmaceutics, I would like now to turn our attention to robotized screening. I will try to give you a short overview of our efforts in this area. In this slide, you see one of the systems that we have been working with. So why should we use robotized screening? Currently, it takes an alarmingly long time to find novel and suitable, sustainable materials. And consequently, there is real urgent need to development and progress in this area. And to meet these challenges, we have for many years been engaged in screening efforts and concurrently pursuing an initiative to develop a novel robotized screening platform to accelerate the discovery of novel and sustainable materials in different key areas. And then by employing a robotized screening, we really can accelerate the identification of islands within compositional and chemical space that exhibit unique and desired material properties. And the areas we have been applying this to is different type of screening, salt screening, co-crystal screening, also reaction crystallization scale-up, but also quite a lot in recent years in material screening. And here on the right-hand side, you can see a previous platform that we have been working with in action. However, this system has several significant disadvantages. It is very expensive, it has high maintenance, and it's actually very space demanding. And to develop this platform further is very challenging. And if we would like to implement AI or machine learning, it would be very difficult. And furthermore, it also generates a significant amount of waste. However, depending on the really explosive development in the field of robotics and AI and machine learning, it's now possible to design much more intelligent and compact, as well as versatile screening platforms that can replace these larger systems with smaller modular systems with AI capabilities. This type of modular system can be adapted for more specific tasks and at a relatively low cost, opening up for exciting opportunities for faster detection of new compounds and in various domains. This type of modular system has also other important advantages, such as much less use of material. Of course, the implementation of machine learning and AI is much more easy, and we could therefore also go forward to create an autonomous platform. And this new system also has very high repeatability and throughput. And currently, we are looking into developing screening platforms connected to precision medicine, utilizing 3D printing, and also traditional formulation of screening. And when it comes to 3D printing, we are particularly interested in implementing automated quality control, which includes imaging and spectroscopy. Efforts to develop the overall new control system for the platform is currently ongoing. And this slide is just to illustrate how schematically, how module-based screening platform can look like. And reaching for a new system, we have, of course, been working with different types of prototypes systems, and this you can see on the left and right-hand side. Finally, I would like to mention some personal insights to what I believe can be done to further accelerate the development and unlock the full potential within the title area, and potentially being able to advance forwards an autonomous laboratory. And I believe that the integration of computational modeling and experimental workflows can streamline the process and guide the experimental design, and thereby reducing the number of trial and error experiments, and thus accelerating the identification of promising candidate materials and substances. We need more tailor-made models for specific systems to speed up the calculations. Many approaches are currently quite generic, which also reduce the precision. Moreover, we also need to facilitate access to critical data information via knowledge bases, and additionally, expediting the validation of new models is imperative to access success here, and we must generate a greater volume of high quality data, which is key to really develop machine learning models. And finally, we need to implement more high throughput characterization techniques, for example, high throughput microscopy and spectroscopy, as well as X-ray diffraction, to rapidly analyze the properties of materials and screen larger sample libraries. I would like to have one takeaway in this talk, and in my belief, it's the importance of being able to integrate in silico screening and robotized screening with AI and machine learning to accelerate pharmaceutical development and advance towards the possible realization of a more autonomous laboratory.
Which of the areas you presented do you believe has the greatest potential in the long run, or in the short run?
I think currently leveraging AI and machine learning algorithms for data analysis, predictive modeling, and decision making for screening processes can really significantly enhance the efficacy and accuracy in the area. So this is happening so much in this area. So I think this is really explosive. And then, of course, advances in robotics and automation technologies will enable very high throughput. So there's a lot of things happening in this area.
Welcome, Swapnil, and please start.
Yeah. Thank you, Anna. Good afternoon, all. Myself, Swapnil Chavan. I am a computational toxicologist at RISE. I lead innovative research in the field of predictive toxicology by using AI, chemoinformatics, bioinformatics, and image informatics techniques. It's my honor to present my team's work in today's webinar. My subject of area will be automation of a predictive toxicology. We all live today in a digital world where we enjoy smartwatches, smart TVs, smartphones. We have a home automation, and a self-driving car is on its way. This digitalization and automation has also impacted our pharma sector. And my today's talk will be centered around automating the field of predictive toxicology. I will be talking about traditional predictive toxicology. What are the obstacles in this traditional way of doing predictive toxicology, and how AI will help us in overcoming those obstacles. So without a further ado, I will move on to my presentation. So I will be talking today about our AI Tox suite. It's an end-to-end solution for automated predictive toxicology with the help of AI. So the main founding stone of a predictive toxicology was placed in the year 1961 to 69, where the field of physical organic chemistry saw a big revolution with the invention of a QSAR. During 1961 and 62, Hanch and Fujita and their team, they come up with correlating the biological activity with the help of few electronic constant and few partition coefficients. Later on in 1969, Hansch has expressed this correlation of biological activity in the form of a mathematical expression, and this was known as a first QSR model. Uh, with the advancement in computer hardware and software, this small mathematical expression is now transformed to a more complex form with more computational power in hand. So this is how in a current uh, world, this traditional QSAR modeling is done. Uh, the chemical structures, either in the form of mole file or SDF file or a SMILES notation, are being used, and then they feed to a software where these SMILES or structures are converted to some numerical numbers, and these numbers are called as descriptors. Afterwards, these descriptors are then further refined. If they are highly correlated, few of them are dropped. They need to be scaled, they need to be centered, and these all experiments are known as feature engineering. After feature engineering, these descriptors are used to fit the model. For example, equation of line. And then this model is used for a forward prediction, either a biological activity or a toxicological activity. What happens is uh, when this main molecule is being transformed to this numerical descriptor, this transformation is not foolproof. There are flaws in this process, as well as when these numerical descriptors are undergo feature engineering, there are few issues. And I will be walk you through with what are the problems this traditional way of QSR building face. The first shortcoming of this descriptor is they can't be derived for a mixture and salts. So let's take an example of a lead acetate. When one need to use a lead acetate for a QSR modeling, they need to convert this structure to a QSR ready structure. And this QSR ready structure can't handle a ionic bond. So your lead acetate is converted to a acetate. And while doing this QSAR ready transformation, you this is a mutagenic structure. You end up converting this mutagenic structure to a non-mutagenic structure. Second drawbacks with this descriptor is the when your molecules have a carbon, hydrogen, oxygen, nitrogen, it's much easier to calculate these descriptors. But if your structure has non-common elements like lead, antimony, barium, beryllium, mercury, chromium, the softwares cannot able to handle this, and a lot of these descriptor having a missing values. So that's the second disadvantage. They can't be derived for a non-common elements. The third disadvantage is while handling the large structure, such as microlite antibiotics. Such a large structure, their descriptor calculation could be difficult and a time consuming. The fourth drawback with this descriptor based QSR is one need to do a pre-processing of the descriptor and a feature engineering like scaling, centering, then removing the highly correlated descriptors, removing the zero variance descriptor. So this feature engineering is an essential step in the traditional QSAR, but that needs a time and a manual steps. The fifth disadvantage is the number of available descriptor are limited. For example, for a numerical descriptor, the AlvaDesk software, formerly known as Dragon, has approximately 6,000 maximum descriptors. And for a binary types of descriptor, there is a Pharma Gobi 2D descriptor, which has close to 40,000 bit string. And this is a limit because in a in a traditional QSR, there is a rule of 1:5. So for 5 chemicals, you need a one descriptor. That means if you have a maximum 6,000 descriptor, you could use this to model up to a data set of a size 30,000 compounds. But if your data set is higher than that, then you don't have enough descriptor to model such a big data. And that's the fifth disadvantage. The sixth disadvantage is sometimes they can be identical for a dissimilar molecule. What I mean by that is here are the polybrominated compounds. And if you calculate a PubChem fingerprints for these 4 compounds, they all those will be exactly same. So imagine if someone is using PubChem fingerprints for these polybrominated compounds, they are representing same compound for a very dissimilar molecules. And that's a big, big drawback. One could argue, okay, PubChem fingerprints are a binary and it could be possible that they can have identical pattern for a dissimilar compound. What about numerical descriptor? Then answer is yes or no. So if one could talk about a single numerical descriptor, let's say a melting point, you can find definitely chemicals where 2 chemicals have a same melting point. 2 chemicals can have a same log P values. So they can be same, but if you look for a set of property numerical descriptors, then they might not be identical. What about in case of tautomers and stereoisomers? Well, in that case, they could be likely exact identical. One need to figure that out. So the identical descriptor is the sixth major drawback with the descriptor-based methods. The seventh drawback is they are non-reversible. What I mean by that is if you have a structure and if you run this through some descriptor calculation software, you will end up having a table of a descriptor, let's say molecular weight, log P, hydrogen bond, acceptor, donor, TPSA. But if someone give you this table of a molecular weight, log P, hydrogen bond, acceptor, donor, TPSA, and ask you what is the original structure, one could not trace it back to the original structure. So they are a one way, they are non-reversible in nature, which is a kind of drawback. The lastly, the mechanistic interpretation. So once someone use this kind of a descriptor for model building, but if one need to interpret the outcome of a model, then they need a domain expert who could able to understand what is the log P, what is the hydrogen bond acceptor, and then he could use his domain knowledge. For example, here, if there are 2 oxygen, and that's why they have a hydrogen bond acceptor as a 2, and these 2 are forming some kind of a hydrogen bond accepting activity within a structure, that therefore it's been seen as active or inactive. So without such a domain knowledge, one could not do an interpretation. So requiring a domain knowledge is another big drawback with these traditional QSR methods. So one could ask, can we just not do without a descriptor? In another way, is there a way where one could do a descriptor free modeling? And today I'm gonna present you what research we did within the RISE, where we have used 2 different descriptor-free approaches to build our in-house models. So representation learning is last decade's biggest revolution we saw within the pharma field, where the graph-based technique and a text-based techniques are very popular. The graph embedding based deep neural networks are the models. They transform the molecules to some kind of a graph where every atom is considered as a node and every bond is considered as an edge. And once this molecular structure is transformed to a graph, then they convert it to some kind of a one hot encoding, which means if it's a yes and no table. If there is a nitrogen present at a position 1, if it's yes, it's given a number 1. If it's no, then it's given a number 0. So that's where we get the one hot encoding as 1 and zeros. Same applies to the edge embedding. If the bond is present or absent, what is the hybridization of bond? So in that way, they given a one hot encoding. And then this one hot encoding is attached to a sequence of neural network layers. And after a model training, one could able to predict whether the given compound is a toxic or not. Uh, Second approach is a text embedding based deep neural network. So in our pharma field, we use a smiles notation as a text. So what it does is it uses a standard library of all periodic element uh, as a table, a lookout table. And then for every character within a smile, we look for this lookout table and assign the given index of the given character. So for example, the C is at the position 1. We transform the first C as a 1. Again, second C, 1. The oxygen will be given 2. The double bond will be given 5. So in this way, we transform this text to a numerical number, and that's known as a text embedding. And then this text embedding is fed to a sequence of neural network layer. Then we train the model, and that way we come up with the probability whether given compound is a toxic or a non-toxic. So these are the two methods that we have, which is known as a descriptor-free methods. And what today I'll be talk about this second method, the text embedding based TNN. So now the first advantage we got is we got rid of descriptor. The second advantage is it provide a ease of quantifying the uncertainty. Uh, in a neural network, it's very easy to quantify uncertainty. The neural networks are a sequence of layer which consist of nodes, and a lot of mathematical transformation happens within the node, which are fed to the next layer until the last layer. What uncertainty quantification looks is it null the certain nodes input and output. By doing that, it stop model to look the same pattern time and again. By nulling the different operation at the various iteration while model training, it does not depend on seeing the same pattern. This is what we call a regularization. And by doing this regularization, while in a in a forward prediction step, one could able to derive what is the uncertainty within the model. So that's the second advantage with this descriptor free approach is they are easy to use for quantifying the uncertainty. The third advantage of these descriptor free methods are so one could use the text to transform them to a text embeddings, and these embeddings then attach to the neural network to predict the toxicity. This process is a reversible. That means one could use certain explainable AI techniques and use the predicted outcome and trace it back through each layer of a neural network all the way up to the input and see which part of input has a highest contribution for the given predicted output. And the explainable AI is a newly developed field which help one to understand what part of the input was valuable and contributing towards the given prediction. It also allow one to debug the model if model is looking at the wrong structure or wrong part of the structure. So this was the third advantage of a descriptor free method. So lastly, I'm gonna show you one of the example from our AI Talks model suite. This example is the mutagenicity model. This model has been trained with quite a large mutagenicity data set come from a Helma et al.'s 2021 paper. We use recurrent neural network, the text embedding best approach to construct the model. And for model explanation, we use integrated gradient method. The model results looks like something this. For a twice repeated tenfold cross validation, model has shown AUC value of 87%. The balance accuracy is 81. The sensitivity and specificity all are above 80%. For a external test set, also models showed quite robust output with the AUC of 87%, the balance accuracy 79, accuracy 79, and sensitivity specificity close to 80%. So was a quite robust model. And then we deploy this model to a web server. And this is how the terminal of a web server looks like. So it just need input in the form of smiles. So let's say this is a cyclophosphamide, and in the form of smile notation, it looks something like this. So one need to just feed in this query to the given section of a terminal and then press run. Once that's done, this is how the model gonna spit out the output. So the output will be consist of the prediction, whether it's mutagenic or not. Then the second information it will provide is the uncertainty, how confident the model is for labeling given input as mutagenic. So here model has shown 100% confidence. And the third advantage is it comes with the explanation. So it shows a toxicophore. So for the given cyclophosphamide, it's an alkylating agent where a phosphoric mustard play a big role. And our explanation has highlighted that these functional groups were very important while labeling this compound as mutagenic. Another important substructures was this nitrogen mustard, the nitrogen with the two halides. And as all of you know, the nitrogen mustard is an important alkylating group, and that could cause a mutagenicity. So this was a very good example how the model not only do the prediction, but it also provide the uncertainty and the explanation why they predicted something mutagenic. So one more advantage of our text-based embedding is we could not only limit to the structure, but it can also be applied to a reaction. So here is a reaction of a cyclophosphamide synthesis. This is a class number one could look for in a SciFinder. So it has this oxazophosphorin derivative along with the dichloroethane, which lead to a formation of cyclophosphamide. What I did in a input is I used this first group, and then with the full stop, we added the dichloroethane as a second group. And using this input, it's a SMRC format of a reaction. Our model has predicted this as a mutagenic with slightly low confidence, but with a true output, that is the mutagenicity. So these AI models are not only limited to a single compound, but they could be extended to a reaction, which a traditional method cannot do. Traditional QSAR can't handle such an ionic bond. They can't handle a reaction. They can't handle a mixture. And the model is smart enough that it knows these are the reagent for a given reaction, and they have a component that could cause a mutagenicity. So model was quite smart. And with this reaction, what usefulness of this new text embedding methods will be, they could be widely used in a life cycle assessment field where there is not one component related to the compound, but there are his pre-formulation components and their degraded product of the given compound. So this text embedding methods has a huge potential playing a role for a life cycle assessment. With that, I summarize our AI Tox tool. So this suite has consist of human toxicity endpoints like CMR, carcinogenicity, mutagenicity, reproductive toxicity, as well as heart, liver, eye irritation problems. Then there are 12 endocrine receptor-based models. There are 3 ecotoxicity related models. So in summary, uh, a QSAR model should be predictive, should be certain, and should be explainable. With our AI Talks models, they fulfill all of these 3 criteria, which make them a good models. The second advantage what our models have is they can be applied to salts and mixtures, which is not possible with the traditional QSR methods. And lastly, our models could be applied to the reactions also, and we have screened those for human and environmental hazards. So we see a huge potential of this, our text embedding based AI models towards the life cycle assessment. So lastly, I would like to acknowledge my colleagues, the Eric Ilippa, who is my former colleague from the Intelligence System Unit at RISE, then Maria, currently a PhD student within the RISE in an Intelligence System Unit, Professor Dr. Ian Cotgreve, our senior researcher at a Chemical and Pharmaceutical Safety Unit in RISE, and Sverke Janssen, the steering committee member of a AI Talks project from a computer system unit in a RISE. I would also like to thank RISE for funding this project via SK Medal funding and the Mistra SafeChem project. Lastly, I thank you for your attention, and I'll be happy to answer your question either now via face-to-face, or you could also email me via this email ID. So thank you. I close my presentation now.
Thank you very much, Swapnil, for a very nice presentation, and actually also very good explanations for someone a bit outside the field, but also very interested in the toxicology part of the drug development is really a showstopper if you do it wrong. So knowing that already from the beginning is really, really good. So thank you very much. So I will have a question to Swapnil to start with, if that's okay. Swapnil, so you said in the beginning here you're doing something new and there is a lack of standard model reporting format for AI models. So what do you think? Can we expect, and when do we expect QSAR models for with the reporting format for QSAR modeling?
That's an excellent question. That's an excellent question, and let me take you back to when the first QSAR been launched in '69, and the QMRF format comes very late in '80s and '90s. So it took 2 to 3 decades for the first QMRF format. At least for AI, I think it won't take this long. It just we need to gather our community together on this topic, and I think the regulatory agencies should take interest in this. They should organize some conferences, some workshops, and have a concrete discussion about to putting the right format in place. So I think a lot of onus to the regulatory agencies at this moment. I hope if they hear us, they should organize, and I'll be happy to be part of such a team of enthusiastic people who wanna put the format in place.
Excellent, thank you. I also have another question which is raised to you, Swapnil, but I think it's relevant to both of you. And it's really here we have new processes, new types of models that you use to optimize the screening and the drug development. But the competences and the capabilities that you are building here, how is it transferred to the students and to in the broader sense? So perhaps that's relevant to both of you, but start with Swapnil.
Sure, I think it should be part of the coursework of a master student, and if possible, also a bachelor student. We are seeing a huge AI revolution, and with the keeping this our with the current development, we need to keep up ourselves up to date. Same with the syllabus of our master students and bachelor students. So I see a urgent need of revising the syllabus for each field. So they should have some chapters within their syllabus about the new AI technology. So as they grow, as they mature with the research, they could able to easily adapt to the new techniques.
And Per, you are also in the academy as well. Do you have any comments to this?
Well, of course, I agree with Swapnil, but I also think that, of course, there's a lot of specialization, but it's also very important with this type of cross-functional work to make this progress, because looking into, for example, robotized screening, there's many different types of knowledge needed to progress, and we also need to talk to each other. So we can't be too specialized, so we can't communicate. So there is another thing to work with. So there is with this explosive development, there also comes up some interesting problems.
That's
Very
true. And this development is not stopping here. So what do you think, Per, would be the challenges and bottlenecks going forward with computational pharmaceutics and robotized screening?
Yeah, I think currently a lot of the work that we're doing is quite reactive, and I would like really us to become more proactive. And this is, of course, a big challenge because it's, of course, more convenient just to wait for a problem to occur. But to be proactive would be really fantastic. Unfortunately, some of the methods are still very computer intensive, and this is a bottleneck in several cases. And I mean, a problem is also that we really need good data to generate good models, and there is a lot of data out, but some data is really bad, and some is not accessible. So there's also some challenges.
And I think time is almost running out, but I was just thinking when you said we need to be proactive, when you say we, do you think we Sweden, we the world, we the pharmaceutical area, or where
I think actually it's unfortunately I think it's quite general in many areas, but looking into pharmaceutical industry, I think there has been some improvement, but we are still quite reactive, and we need to become better, I think.
I have no further questions. Any more comments you would like to have, or has there come up any additional question? So with that, I think I close this webinar and thank everyone for listening and presenting.